Sorry about that quick interruption there. Okay, so I've got my big beaker filled with water. I've got my light. Do you see how I've kind of balanced it on this little tiny beaker? It's just nice. I've just got a little beaker in here. If the light is not shining straight up into the air, the problem with the light that students will run into is that the light is hot. And so if their plant, if the air in the tube is being exposed to that light, the air is heating. And so it's going to expand and push water up out of the tube that has nothing to do with photosynthesis change. It has everything to do with temperature change. And so the farther away we can put these, right, so they're not being affected by the heat, the bigger this beaker, the lower the light is shining, so it's shining through the beaker. In fact, I even want more than this. If it's shining through the beaker, we're going to have less chance that we've got heating going on. We still even have quite a bit. And the other thing that they can do is actually fill the beaker that the plants are in with water, right? So that there's no, we're minimizing the amount of temperature change gas change due to temperature. Okay, so it doesn't have to be all that close. They just have to be exposed to the sunlight. And they're going to wait 10 minutes and get a baseline rate, and they're recording it here, setup 1 and setup 2, under number 5 of their student guide, what the millimeters per minute are. So they're going to do it for 10 minutes, see how many millimeters it went, divide by 10, and get a millimeters per minute. So each of their plant setups is going to be a little different depending on how much Alodia leaves are in there. So once they have a baseline, then they're free to test whatever they want. And so, also, anytime the water makes it to the top and starts dripping out, obviously they need to start over. So they just uncap and recap, and then measure it again as the first measurement and the second. They can uncap and recap as much as they want because you're not changing the actual plants inside of there. Okay. So then at number 4, they're going to start testing things. Now I would encourage you to have the students grab their little whiteboard at their table and record their results in some kind of display fashion on their whiteboard, so that when we come together we can all talk about it and they can hold up their data. Okay, so have them do it on the whiteboard. And then they can try things. So one of them, the first one, they're actually going to wrap in foil. So one of their setups, they're wrapping in foil. And I had foil, but I've misplaced it somewhere. And so they're just going to wrap the entire tube with foil so that it cannot photosynthesize. I mean, it cannot get any light, right? And then they can just either put it back in the beaker or put it in another beaker and set it off to the side. So one of them is going to be in foil. And the other one, they're just going to start manipulating. So here are some of the things they can manipulate. We talked about it up on the board, what things they can manipulate. And so one of the things is the color of light they receive. So in order to change, I'm just going to pretend this one's in the foil for now, so let's move it here. To change the color of light they receive, do not put the food coloring inside with the plant, because then we can't reuse that plant. It's been ruined. And so it'd be nice if we could reuse our sprigs. So after your lab is done, have your students put the sprigs back in the beaker at the center of the table, and other students can use the sprigs. There's no reason not to. So to change the color of light they receive, you're actually going to change the color of light that's coming into them by changing the color of water around them. So if I wanted to try green light, for instance, I would simply put green food coloring into the water around them. Okay, and I'm going to use the plant to start up. Okay, so can you see with that setup, the only light that that plant is receiving is the green light that's coming through there. Okay, and so we're going to measure that for 10 minutes. 5 minutes, my bad. So we're actually going to do the experiments for 5 minutes so they can get more in. So you can do 5 minutes and you look at how many millimeters. So again, they're going to measure, okay, where are we right now? We're right there, so we're at, see, we've already gone up quite a bit. So we are now at 140, so we went from 119, I'm sorry, wow, I can't even read, 104, from 109 to 104, so we're moving. And then they'll give that 5 minutes and see how much it moves again. And so we're seeing if we slowed it down or we sped it up. Okay, so the independent variable is what they're going to write, green light. Initial rate of photosynthesis, that's their control rate. And then the final rate of photosynthesis, what was it when I manipulated this variable? So the conclusion is green light hurts them, or green light helps them, or whatever, okay? And then the same thing with the foil. At the end of all their experiments, the foil, and they need to time it, how long it's been sitting in the foil. They need to look at the rate there. Now, interestingly, the one in the foil, what's going to happen to it is it's going to stop photosynthesizing. Now, you're not telling the students this. They need to come to this conclusion on their own. It's going to stop photosynthesizing, so you would expect the water not to rise anymore, right? But it's also going to continue to respirate. So it's going to start sucking oxygen out of the air and the water as it respirates. So you might actually see the water level drop a bit back down because it's sucking air out, right, as it does its thing. Okay? And that's what it asks them in 5 and 6. All right, so that's one thing you manipulate is the color of light. And they can be creative. We only have 4 food colorings, but they can use the little recipes on the box, which don't seem to be there. You can go online and figure out different colors, all right? So they can do all different colors. And what they should find, and now in the past experience with this, green light should slow it down because plants don't like the green light. But all the other colors, it's all over the place. So if they use like dark blue, they'll be like, oh, blue slowed it down. But another group who used a light blue, it stayed the same or it sped up. And that's just because of how much light is penetrating the plant. So technically, any of the colors, if you go down from white light, any of the colors are going to cause it to slow down a bit. But it's not a super accurate setup. If you lose the seal, it might change a little bit. So, but that's something to discuss with students as they're presenting their results to each other. So after everybody's had a chance to run 5 or 6 experiments, then you're going to have a discussion like what things helped it, what things hurt it, why. And if we have discrepancies, then you might ask them, okay, so why would you find that blue light did help it and I found that blue light hurted it? Hurted it, oh my gosh, hurt it. And so you can have a discussion about maybe our blue light was a different color. Maybe we lost the seal, maybe there's experimental error, right? And so yeah, you can have those discussions, but what we should find is that the color of light matters. The white light is the best, and that's because, and let's see here, let me make sure. Yeah, so this is when you're going to bring up the photosynthesis, I believe, yeah, the photosynthesis PowerPoint on the projector. And the very first slide, and I'll send this out to you guys. The very first slide is the light spectrum. And so after you've had this discussion and everybody has shared their results, then you will talk about the different colors of light, right? That plants absorb in a certain spectrum. And the way that they figured this out, if you're looking at the slide, actually I might put the slides up, give me a second.